This is a production of Cornell University.
And thank you again for coming. So today I'll be presenting about applications of multi-trait and multi-kernel models for genomic selection in cassava. And I'm really grateful to a lot of people who have helped me to reach this stage. Okay. Well, I'll skip the outline and I'll tell you a little bit about cassava, which is kind of very exciting to me. Cassava is no longer an orphan crop. About 263 million metric tons were produced in 2012. Cassava is a starch machine, so we have about 90% of dry matter as carbohydrates, and then some traces, other stuff. Cassava is an outcrosser, producing male and female flowers from the same plant. And the interesting thing is, even with low inputs and marginal land, cassava is still giving some tangible productivity. So it's very interesting. This is the most interesting to me. It's 2016, I guess. We were in China and we collected this information that China has bought about $1 billion US worth of cassava chips. And that's very interesting. It's gonna drive the market. And I think, again, the reason is that cassava starch is very, very appealing to the food industry. So with that, I make a jump now to my introduction to mixed models. So I was introduced to mixed models by working with Denix at academia. He just walked in. And I learned a whole lot from Denise doing this. So EMM-REML has 3 solvers. The first solver is a univariate single kernel solver. By kernel, we mean one random effect. And it's based on the EMMA algorithm. The second solver is the multi-kernel solver, which is in fact a wrapper on this, and it's based on kernel averaging. And then another interesting solver we have is this multi-trait solver, which is based on the ECM algorithm that was developed by Zhu and Matthews, I think 2014. So the interesting thing about EMM-REML is that it is very flexible and you can adapt it to various things. And we have actually adapted this multi-trait solver to different kind of models, which I will show today. And EMM-REML has in fact been successful in plant breeding literature. So the talk is structured as chapters in my thesis. And the first chapter, we looked at accuracies of univariate and multivariate genomic prediction models in African cassava. So our motivation for this was to understand which models are we to use for GS in cassava. And then the next thing is to evaluate the breeding values from these models and see what's going on. And so for this, we had a data set, an interesting historic data set. It was a multi-year, multi-environment trial data. It was collected from the so-called genetic gain at IITA, Ibadan, Nigeria. And over a span of about 16 years, from 2000 to 2015, we chose these 3 locations, Ubiaja, Mokwa, and Ibadan. And in fact, these locations are very divergent. This is in the rainforest region. This is closer to this, and this is like a semi-arid location. So they are very divergent, and they represent areas of cassava production in Nigeria. So we used what we call clonal evaluation trials, data only. And in cassava breeding, clonal evaluation trials are where we impose heavy selection on the breeding germplasm. So CETs are mostly augmented design with checks, and there are no replications. And this is because of the cassava biology. We plant in stakes, so you can't get enough stakes to do replication. So we fit different sets of models. In the first scenario, we fit models using data from a single environment, but multiple year data. In the second, we fit multi-environment trials to capture G by E. And we use GBS matrix data to calculate the genomic relationship matrix from about 173,000 markers. So this is a little data on the traits we are dealing with and some metrics. So this is the number of records of the data we have at different locations, and these are the number of individuals. And we had about 324 individuals across these locations. So our targets are these traits, 6 traits, seedling vigor, number of roots per hectare, fresh yield per hectare, dry matter, of course, and then mean CMD. This is a very critical trait for cassava, and then mean CGM, which is a pest. CGM is a pest that eats up the stake in cassava. I don't really know very much about it. So in scenario 1, we fit 2 interesting models. The first model is the basic G-block model we call UT, and this is a single trait model. And then the genomic effects are coming from this distribution, given the genomic relationship matrix. In the same vein, we fit a multi-trait model, which is like this. And in this case, we have a matrix of traits, but then the distribution is different. And this is a matrix normal distribution. The matrix normal has a row covariance and a column covariance. And this is the covariance that captures trait by trait genetic variances and correlations. And this this is in fact the eigen decomposition of this matrix, the genomic relationship matrix. And it's a simplification of the algorithm. So to simplify the algorithm further, we use the Kronecker product and then transform this to the multivariate normal distribution. And the Kronecker helps to get this working. But in this case, the Y becomes a long vector of stacking the traits together. Okay, so a couple of things here is that Y is a stack of many traits, and the X, which is our fixed effect, we use location trials nested within year, nested within locations as fixed. And in fact, the algorithm we use can allow you to fix different traits, different effects, in this X matrix. And this is how it works. So we fit this model with the ARML F90 package, which is a member of the family of the BLOP F90 program developed by Mr. and Co. And we go forward again in the next scenario to capture G by E in this data. And we fit, again, 2 models. One is univariate and another is multivariate. And this univariate is an interesting model, which is a multi-kernel model. The first kernel captures the genomic effect, and the second kernel captures the clone by location effect. And in fact, what we do here is that we use the genomic relationship matrix for each location, each individual in each location, to capture the genomic part of the interaction, the clone by location interaction. In the multi-environment model, we use the same trait in different locations. It's just like the multi-trait model, but there are some subtle differences. One of the differences is coming from this VG matrix. In this matrix now, we capture the genetic variance of the same trait at different locations in the diagonals, and then the off diagonals, we capture estimates of G by A as, as the covariances. So the GEBVs from this ME model contains both genomic and G by A parts. And the VE is diagonal. By diagonal, we mean we just allow for variances, variance of the traits, error variance of the trait at different locations, and we don't want correlation between these because there is no mechanism that generates correlation between a trait at different locations. So to match this, to compare this UE model with ME, we join these two together, the genomic part and the interaction part as the GEBVs. So to compute the accuracy, we fit a simple model that we call performance genetic performance model, and this model just is like the first UT model, but in this case the difference is that it assumes an identity, which means that there is no relationship between the individuals concerned, and we fit this in different locations, and we think that this captures the estimated total genetic value of the individual. So here I show results, and in each block is a different trait. So here is vigor, here is CMD, here is root number, fresh yield. In each column is a different location, and the first location is Ibadan, the next is Mokwa, and the other one is Ubiaja. Excuse me. So in red is the prediction accuracy for the UT model, and in green is the prediction accuracy for the MT model. So we see that in most traits, actually, that the, the accuracies of the MT model are higher. And in fact, we see this in almost all the traits. And what we make of this, I show you a table of one location, and this location is Ibadan, showing the heritabilities in diagonal of these traits, and in the off diagonal are the genetic correlations for these traits. So why do we see these accuracies there? We see here that these traits are low heritability traits. We have a bunch of low heritability traits, and then we have this high heritability and maybe these medium ones. And in fact, when you jointly analyze traits of low heritability with high heritabilities, and you have a substantial correlation going on here, we expect that the multi-trait accuracies will be better. Okay, so we shift gears to the second scenario, which is the UE and the ME models. And just like before, we have blocks as traits and then columns. And in red is the UE, and in green, again, is the ME model. But in this case, we see that most of them are neck to neck, in fact, and just in slight places, you have the ME a little ahead of it, higher accuracy. So we get some interesting thing in the genomic metrics in the G metrics of the, I call it VG here, of the ME model. And this is somewhat kind of a description of what this is. And if you think about, for example, vigor here, here in Ubiaja and Mogwa, this means that if we select some lines here in Ubiaja, we expect about 39% correlated response to selection in the next environment. And we see this in all the traits. And in fact, this gives information on a view of how to see G by E. And then something interesting again is that you can, if you have several environments, you can begin to cluster this into TPs. Okay. And this is a table that shows variance explained by the UE model. This is the clone by location. Variance is explained, and this is for clone. And I just wanted to point out here that these U traits have so much G by E variances here compared to other ones. And we know that these traits are so much affected by the environment. And the same thing goes for these. We see here we have lower correlated response to selections. So in summary, what did we get from these? We get that the MT model offers more accurate breeding values for a single location data. And we see that this comes from significant correlations and joint analysis of low and high heritability traits. And we know that these low heritability traits benefit more from this analysis. And then we also see that the ME model and the UE are not significantly different, actually. But we recommend the multivariate environment for this because we see we get a lot of information from both models. Sorry. So something interesting again coming from the genetic correlations that I showed you is that there is good prospect for concurrent improvement of these traits in cassava. And in fact, these are the most, well, I would say these are production traits in cassava, really very important traits. We also see something interesting is that CMD is negatively correlated with other traits, which is the direction we want them to be. So in the next chapter, I'll move to comparing selection gains from multi-trait indices from these models, and it's kind of an effort to evaluate more the breeding values that we got from these models. So in literature, we see that a lot of models are usually evaluated by prediction accuracies. There's a few places where the evaluation is selection gains. But in fact, this is because it's not an easy thing to actually do that. They are predicted gains. So we actually want to know how these models perform based on predicted gains. And one of the problems is having to make these models comparable. It's not an easy thing to make. Even if you have independent data, you should have a comparable reference framework, and you think about which model do I use to make the framework? Is it a multi-trait or a single trait? Anyway, we use multi-trait selection indices in this case to achieve this goal, and we compared also, like before, the UT, MT, UE, and ME models. But we added two other models to these, which I will explain in the next slide. And these models are, if you recall the UE model with two kernels, we have the genomic part and the GE part. So the UEA is just capturing the genomic part and leaving out the GE part. And this is just an average of the entire environment. And then we fit another model, which is MT-ME, which is like a merging both ME and MT models. This we think is an interesting model. The two reviewers pointed out that why don't you do this? It wasn't an easy thing to do. It wasn't an easy thing to do though. But we fit, it's just like the multi-trait model, but in this case, we have 18 traits. We organize this trait as trait 1 in environment 1, trait 2 in environment 1, and in that direction. So we had altogether 6 by 3, which is 18. But then in the error part, we just use kind of the VE from the ME model, which is from the MT model, which allows traits to be only correlated in the specific environment where they are. And this model is complex to fit. It was fit in the Gibbs sampling algorithm of block F90. So how did we estimate gains? So first of all, we standardize all the trades so that everybody has the same stat, and we use the same economic weights. These are the weights, and these weights represent 20% increase in vigor, 35% increase in root number, 45% in dry matter, 35% decrease in CMD, mean CMD, 45% increase in yield, fresh yield, and 10% decrease in MCGM. So the index was calculated as this. So matrix multiplication of the economic weights with the GEBVs from each model. We selected the top 65 clones from each multi-trait index. We estimated gain as the mean of the index values of these selected. And the trait gains we estimated as the mean of the GEBVs of the selected individuals. So here we have a plot. This is Ubiaja, this is Mokwa, and this is Ibadan. And we have this box plot. I want you to notice something that in all the locations, even here where we have something like a tie, the MT model is doing better than the UT model. But we see here that the UE model is doing much better in Ubiaja, and then also slightly better here on average in Mokwa, and slightly better again on average at Ibadan. Well, let's jump into the traits and see what was going on. So here is Ubiaja, and each block is a trait. This is vigor, this is root number, this is dry matter, this is CMD, this is fresh yield, and this is CGM. And again, they are colored as the same. This is UT, MT, UEA, UE, ME, and MTME. I would want to point out that this UE is more like what we do in the next gen, is the selection index we use in the next gen program. So we see here in Ubiaja that the UE model was doing well here. And you come to root number, which is a trait that has so much influence by the environment. The ME was performing better. And in dry matter, the ME was on average slightly, the ME and the UEA were doing well. So I point out here in CMD where the UE model was really dominating others. We want this to go down. So this going down is a good thing for us. So this is a place that this model is dominating all these indices. And then for fresh yield, it's doing pretty well. And these models rank almost the same in this case. So we move to Mokwa, a semi-arid region, and we see the MTME is doing much better here with vigor, root number. The MTME is still doing better. It does very bad job here for dry matter. Again, the UE and the UEA are dominating for CMD here. And in yield, again, this MTME model is dominating. So we move again to Ibadan, and again, we see the MTME, the ME model is kind of dominating here on average, and in root number is the MTME. Here I can rank these models 1, 2 as almost same. And then again in CMD, the UE model is dominating in all locations. So in yield, the MTME again, and in CGM, the UE is doing very bad here. Actually, they should be going down. Okay, so what do we see from here? We see that some models have higher GABVs than others. And we kind of ask a question, since we are using breeding values, can we combine breeding values from different models together? Well, why not if we are interested in making gains? So we try this, we replace the breeding value for CMD from ME with that from UE. And for dry matter and CMD at NTME in MOQUA, we replace with UE and UEA. And the same here for NTME, we replace with this and this. And then we move on to see what happened. In red is this composite index, I call it, and you see it becomes superior to other ones. So I know that there can be a lot of arguments on why this is not a good system to evaluate these models. There should be a separate independent reference to do this. But this is just kind of showing a clue of what is going on in this. And I think an ideal case would be to have independent reference data to be able to evaluate these models or GABVs. So from what we learn, we see that accounting for G by E is very important for making gains in cassava. Because of G by E, we probably need to use several models. We don't know which one would be good. And we also, in this case, recommend the multi-trait models because they have a lot of information, multivariate models, sorry. They have a lot of information. And this shows good prospects, actually, for cassava improvement. The composite index may also be beneficial. Okay, so now I make a jump to something else. A GWAS type, like type analysis, which is mapping traits. So we have an issue in cassava breeding. We are trying to move towards this beta carotene cassava, which is sponsored by Harvest Plus and so on. And we call it so-called yellow cassava. But we have a challenge in yellow cassava. The challenge is that most of the yellow cassava have low levels of dry matter, actually. And we need to understand what is going on in this. But we know already that there is a negative correlation between dry matter and tuber yellowness in cassava. So we set out to understand the genetic basis of this. And here I show a plot. This is the white cassava part. This is the yellow part. And this plot I borrowed from Ismail Rabi, which was just plotting dry matter against yellow coloration in cassava. So here you can see clearly that these populations split. Okay. So we developed this so-called regional heritability mapping procedure for cassava. And this procedure is a very interesting application of linear mixed models. So what we do, we divide the chromosomes in cassava, 18 of them, into windows, sliding windows. And these windows are made up of 100 SNP each. We call these segments. So we have this window sliding through, and there is an overlap between them. This overlap is 50 SNPs adjacent to 50 SNP overlapping with adjacent windows. So we make a genomic relationship matrix from these segments. We make another genomic relationship matrix from the whole genome SNPs, and this is to correct for background effect and population structure. We fit two models, one null and alternative model. Show here, the null model is just like the G-block model, using one kernel, using all the SNPs that we have. But in the alternative model, we fit two kernels, one kernel for the genomic segment we want to capture, and the second kernel correcting for background effect. So we use likelihood ratio test to test for significance of this kernel. And we plot this as Manhattan plots. Excuse me. So what data do we use? We use the so-called genetic gain data again, but this time only measured from 2013 to 2015. We use a bivariate trait called pulp color, which just is, a binary trait, which just measures whether you are yellow or white or maybe off-white. That's the case maybe. So we use this to separate the population into two parts. We use GBS SNPs numbering about 177,000, and the number of the whites were 451, and for yellow were 262. So phenotypes, of course, were from 3 locations again, but we corrected these as fixed effects. So here we show the LD between segments. Each segment in a chromosome, and each color change is a different chromosome. So here we see the kind of LD we get. We have this low LD giving us confidence that when we get associations, these associations may be real. Okay, here we see this length or length of genomic segments in base pairs, and we see that a lot of segments are between 300 plus 1,000 kb. Yeah. Okay, so when we plot the results here, we see we see in the first plot is the white cassava, and in the second is the yellow cassava. So we see here we pick up signals in chromosome 1, we pick up from 4, from 5, from 10, from 17, and from 18, and then in the yellow we pick up only for chromosome 1. And so bear in mind here also that we have like very little sample size. So what next do? We try to see whether we can extract some information from the genome by doing candidate gene analysis. And we extracted information on 500 kb flanking each side of the heat we had. We'll show later the choice of this 500 kb. And based on literature review, we were seeing 2 families coming up all the time. And this is glycosyl transferases, and digging in deeper into this is like carbohydrate enzymes, of which starch synthesis, sucrose synthesis, and so on and so forth are in there. And we see another family of serine kinases, and this family regulatory protein kinases. We also see invertases, both neutral and acid, and some other plant regulatory regions. So we also looked on literature, and we saw that these serine kinases already have transgenic studies which are implicated in starch. So before I go on, I want to point out that our goal was not really to say that this gene is what is causing this, but we kind of think that these genes may be tagging regions which are very interesting for dry matter in cassava. Okay, so how do we validate this? Then we performed a validation. Excuse me. We performed a validation for this serine kinases. We went to the genome and we grabbed everything called serine kinase, or looked like it, and we extracted about 1,200 genes. We encoded it to about 7,000 SNPs, and we used an independent population, which was the progeny of the genetic gain, 100 genetic gain parents, to validate these genes. Excuse me. We also took randomly genes from the genome, about 2,000 genes, and anchored about 7,000 SNP as control for this. Now for this we fit this 2-canal model again, In the first kernel is this target, which is the serine threonine kinases, and then the second is every other thing, every other SNPs in the genome, not this. And we get prediction accuracies for this. Okay, so we do this as well for we had this Saiton paper which reported cassava genes involved in starch biosynthesis. So we took those genes, excuse me, and anchored them to SNPs. We call them cassava starch genes. We also extracted all things called genes in the RH, in the significant regions we had. And this gave rise to about 2,300 SNPs. We call them region genes. Now, based on literature, we had this 53 list of genes that we saw that we could be implicated in starch. And we also got these and anchored them to SNPs, about 400 SNPs. We got another 53 randomly just to control for this. And then we went into this region and said, oh, well, let's get those genes that are in this region that are not reported in literature, just to check what is going on. And we also anchored them to about 300 and something plus SNPs. So we used these as well in the same methodology we used for the SNRKs. So here we show the prediction accuracies for the snacks, for 7,000 plus genes. It was about 50% of the prediction accuracy we get from the entire SNPs in the genome. So others were about 20 something plus %. And, and the random part for the snacks was about half of what the snacks had. So interestingly here, when I do the anchoring of the SNPs, I find out that all the time the same SNPs come up again and again, which means that we are really tagging something in the regions. Okay. So another thing we did is that we kind of, since Saiton has already published these genes, we wanted to see which genes which in our region that are intersecting with these ones that Saiton picked up. And we saw 4 genes, 2 of them, one is an acid invertase, one is a neutral invertase, I think so. And there's this phosphoglucose isomerase, and an interesting one, the fructose bisphosphate aldolase. So a quick search on these shows that Monsanto has a patent pending on this. So again, we want to point out that we are not searching for functionality of these genes, but we are tagging regions in the genome that may be implicated in dry matter. So this is just a plot showing all these genes we had and the regions where we kind of saw what was going on. I'm sorry, I don't see very much. Okay. And this is in chromosome 1, where we had top hits and the proximity of different genes that we saw we selected as candidates. Again. So how do we validate the RHM procedure itself? We use the so-called high-causal SNP methodology. So we move into the genome, we select 5 adjacent segments by random. And on the 3rd segment, we choose a SNP by random and inflate the variance of the trait to about 10% by adding effect. And we make G matrices for each of the segments. But when we come to the 3rd segment, we remove that SNP that we used for inflating the variance, phenotypic variance. We run this several times to see what was going on. So this gave a clue on the span of our candidate gene analysis, because we could pick up genes about 2 segments. We could pick up effects about 2 segments away from the place where we added this effect. So we saw that the RHM was a good procedure for polygenic traits. And in this analysis, we picked out that 102 times out of 216, we picked the effect that we added. And this was about 47% power of detection for association. And we think that with increased sample size, we probably will benefit more. So in summary, the RHM is informative for mapping complex traits. Genomic segments may be employed in breeding. There needs to be some research to do this. And we see that there is good prospect for improvement of dry matter, especially in white cassava. And then dry matter as well is a polygenic trait. Okay, so in case you have not seen yellow cassava, this is how it looks, and this is white cassava. Okay, so I move on to the next chapter, which is moving on from the regional heritability mapping for one trait and trying to map two traits at a time. And in this case, we want to see how two traits are co-inherited together. And here we use dry matter, again, the yellowness, which is root color, and then we also use fresh yield in some trials that they call high starch trials. So our motivation is to see how dry matter and this yellowness are inherited together, and again to see whether we have like prospects of developing yellow cassava. So how does this go? The RCHM procedure starts by fitting. I would point out here that the ideal procedure should have been exactly what we did for the regional heritability, but in a bivariate model. But we tried to do that without success because I think probably we have so much less data. We could not estimate it. So the models were not converging. So what we did was to look for another procedure to do this, and this is the procedure. So we fit a bivariate G-block model where we use the MT model, as we showed before. We use stem principal components in the fixed effect part. And before we did this, actually, we brought out these 10 pieces and we regressed them to dry matter for a start. And for white, we didn't see much value for the pieces. They were not significant. But for yellow, we saw that these 10 pieces were explaining about 23% of variation going on. So it was essential to add that. So after fitting this bivariate model, we move forward immediately to calculate SNP effects from the model, and this is done very much by animal breeders, so-called SNP lab, and you just back solve using the estimates that we got for the breeding values, the genomic relationship matrix inverse, the marker matrix, and this matrix may be used to probably weight SNPs if you are interested in that. Okay. So after solving for SNP effects, we divided the genome on each chromosome into 3 fairly equal parts. The mean of each segment was about 18 megabase approximately. So what we did was we calculated so-called genomic segment values from these, excuse me. And genomic segment values were the marker matrix for the segment we're in multiplied by the SNP effect in the segment we're in. So these genomic segment values were calculated for both traits, and then to get a correlation, genomic segment correlation, we just correlated this genomic segment for the 2 traits we're interested in. So in fact, this is a genetic correlation because we are correlating sort of, this is a proxy, looks like a breeding value. But okay, so we did this for dual traits. We're interested in dry matter and B, dry matter and fresh yield, and so So what data did we use? We used the same data we used for the RHM. We applied these to the same populations again, white, yellow, and in this case, we also joined them together, we call yellow plus white. Okay, so we applied also for dry matter and fresh yield in so-called high starch white trials. And these are trials that had high levels of dry matter and good yield, and they were planted sort of to understand when best to harvest. And one set of harvest was done 12 months after planting, and the second set of harvest was done 14 months after planting. So we plot these segment correlations genome-wide to see what's going on, and excuse me. And then we also try to say that when these segment correlations are going in the direction that the breeder wants, then they are favorable for what you're looking for. We use so-called residual resampling for significance testing. Okay, I show here a plot. Each bar represents a segment. Change of color represents another chromosome. And this is for white. And we see that most of the segments are in the positive direction. And in yellow, excuse me, in yellow, we see that most of the segments are going towards the negative direction. This is for white mixed with yellow, where we see a mixture of what is going on. Okay. So we see 70% of the segments in white were favorable. And in fact, from the bivariate model, we saw that the genomic, the trait correlation was 0.14, which indicates progress can be made using a multi-trait index. So we move over to yellow, which is really more interesting. And we see that 81% of the segments were unfavorable, and the whole genomic correlations were negative 0.3, indicating that developing yellow cassava could be challenging. So interestingly, this doesn't happen in the South American germplasm. So Ceballos has this publication and has been saying that just a recurrent selection was doing very well for both traits in South America. Okay, so we argue here
This has been a production of Cornell University, on the web at cornell.edu.